Hello, everyone, and welcome to today's National Microbiome Data Collaborative webinar. We are an initiative supported by the US Department of Energy aimed at developing standards, processes, and infrastructure for an integrated data ecosystem. We support data integration efforts and enable science by connecting data, people, and ideas. Today's webinar will showcase the new features of the NMDC submission and data portals to support the DOE's science focus area projects. I'm your host, Jonelle Basso, and I'm a research scientist in the Plant Microbe Interactions group at the Joint Genome Institute, and I spearhead engagement strategies and activities with the NMDC. This event will be led by NMDC co-PI and submission portal lead Montana Smith and data portal lead Alicia Clum. Montana has worked in sample data and metadata management since 2015, and as the EMSL submission portal product owner, works to improve sample metadata collection and to adhere to DOE FAIR data standards. Her role as the EMSL metadata lead and her involvement with the NMDC ensures that relevant EMSL user projects will be interoperable with the NMDC requirements to improve data sharing. Montana also engages with users regularly to improve and expand metadata collection. Alicia is the Microbiome QA/QC manager and product owner. She has worked on a wide variety of genomics and metagenomics projects and is responsible for overseeing NMDC's portal workflows and corresponding documentation, assuring data generated by NMDC workflows meet quality standards and that there is increasing use of NMDC's data portal. So in brief, today's webinar will follow this agenda. Montana will lead us through what is the NMDC and several interesting aspects of the submission portal. Alicia, in turn, will touch on NMDC data access, discussing elements of the data portal, API, and the new K-base lakehouse. We'll also have some time for questions at the end. To help facilitate Q&A, we'll be sharing a link to a Google Doc throughout this session, where you are welcome to add questions either to the chat or Google Doc directly. So without further ado, we welcome Montana to get us started.
Thanks, Jonelle. Okay, so before I kind of jump into NMDC and everything else and all of these details, I want to kind of set the higher, bigger picture, higher level stage for us. So microbes are everywhere. They're fundamental to life on Earth. They play a very vital role in every sector of biological science and have strong applications for biotechnology and bioeconomy. I probably don't have to tell all of you that, but still nice to set that picture. The Biological and Environmental Research Program within DOE has made large investments to better understand microbes and the microbial community functions and how they apply to the broader mission of developing predictive understanding of Earth systems. And as this field expands and the variety of data and data types becomes available, the management of that data for sharing and reuse is becoming fragmented and inefficient for an ecosystem scale evaluation. So NMDC conducted a survey about data management and data reuse, and we uncovered that some of the key challenges were a lack of metadata, a lack of standards, and a lack of data access. And so the NMDC was initiated to address some of these problems and develop infrastructure and tools to improve data reuse and advance science. But now, 6 years later, we are seeing AI application across the biological environment and with research. But we have the same challenges for AI readiness and for AI data access and reuse. And the solution and the needs are still the same. Okay, so NMDC provides the microbiome data integration hub focused on providing an infrastructure for sharing data and solving problems. We have developed infrastructure to share critical data and metadata for describing experiments and to enable interoperable multiomics data that is important for understanding these systems. This is accomplished with our core capabilities of standardized metadata capture via our submission portal and data exploration through the data portal and data access via the data portal or API. So I'm going to focus on the schema and metadata submission side of NMDC and specifically how we are leveraging AI to simplify the process of metadata capture and applying standards and adhering to requirements for data generators to also provide AI-ready data, not just findable, accessible, interoperable, or reusable data. The first step to simplifying metadata capture is to provide a one-stop shop so people don't have to get familiar with different standards, different submission methods, different systems. So NMDC is very tightly coordinated across BER programs to improve this process of metadata capture. And specifically, we work really closely with the JGI and EMSL to provide a single place for submission for researchers and for capturing that metadata. The NMDC schema provides the flexible but adaptable backbone to this metadata capture and all of NMDC's capabilities to describe multiple experiments, types of samples, different data types, all of the spans for this microbiome science and expanding for BER. The submission portal is schema driven and provides tools for metadata capture. And NMDC has spent a lot of time addressing these key challenges with standardized metadata. So, of those key challenges for data reuse, focusing on the metadata aspect, capturing complete and structured metadata is hard. And it's hard because there are a lot of standards that you could follow. And even when you choose one to follow, it's not always interoperable. It's not always aligned with other requirements from other repositories or data sources. Here I'm showing an example that we queried from NCBI where, very clearly displaying that applying standards is hard. If we look at metadata term depth, which would seem relatively straightforward at a high level of just number and value, but when you start to dig into how this metadata applies to different types of samples, it becomes a little more complicated, or maybe the guidance isn't necessarily totally clear for how somebody might want to capture this metadata or what this field is for. Similarly, even when you have a metadata standard and when you just capture the metadata according to that expectation, there's not always enough information there to make data truly reusable and provide the critical context to understand an experiment and reuse data. And this is the gap that NMDC has been filling. And we see this gap as a problem whether we're talking about a human researcher or AI application. And so as we charge ahead, I will be presenting today how we are leveraging the power of AI to improve metadata capture and make the overall process of capturing this information easier for data generators, while also applying AI to make this process easier. Things keep looking weird. I mentioned interoperable metadata across BER programs on the NMDC submission portal. So when you come to the submission portal to capture your metadata, we first ask questions about the data that has been generated. You can submit to NMDC regardless of whether data has already been generated or if you're just starting an experiment. You don't have to have a user project. You don't have to have a project that's associated with DOE. You can use the submission portal to capture your metadata regardless of where your project stands or where you're at in the data lifecycle. Here I'm just highlighting again that we do support the two user facilities, JGI and EMSL, for metadata submission. So you can do this all in one place. When completing your NMDC submission, we ask for information about the study that the data that these samples were generated are related to and links to any publications or funding source or DOIs. And later you'll see why this part of the submission is important for the AI tools that we've implemented. Before jumping into those metadata tools, I want to add that we received a lot of feedback from submission authors that it is common within a research project for the individual that has information about things like funding source and study details to be different from the individual who knows information about exact samples and metadata details. Or somebody might receive samples from an external source like Neon, and so they may not have all of the metadata requirements and the details that are being asked for. And so we've implemented this contributors role and contributor section within a submission. So multiple people can make edits to a submission. The initial author of a submission will just need the ORCID of the individual that they wish to give access to, and then allow that individual editor permissions for the submission. So providing this collaborative connection within science. Okay, so getting into the fun part, the actual metadata capture in all of our submission tools. I'm not going to spend a ton of time here because we have a lot of submission tools, and I could spend almost half an hour just talking about each of these tools that we've rolled out within our submission portal to make this process easier. We have really detailed documentation that you can reference in our technical docs. Just kind of a quick high level, we have in-sheet validation. We provide guided enumerations for metadata that follows metadata completion guidance and requirements. We provide a term search. So if you don't know where your metadata field is, you can search for it there. And readily available metadata help for the metadata terms that are being provided and asked for. But so those are some of our initial tools that we've had rolled out for a while now. And for now, for this webinar, I'm going to focus on the metadata suggester tool. The metadata suggester tool recommends metadata based on other information within a submission. This functionality is accomplished through two sources. It can leverage existing data via an API call or some other data source and recommend metadata for your submission. So the example that I'm showing here is referencing the submission, what the submission author provided, the latitude and longitude. By providing latitude and longitude, the suggester tool can ping Google Maps API, and it provides a recommendation for elevation metadata. It's a relatively simple metadata field, but it's one less requirement that somebody has to manually fill out or has to think about the correct structure. You can see here that elevation's required in meters. And so if you use this tool, you don't have to think about whether or not your source is in feet or meters. With our eyes again focusing on AI and advances in AI, we asked ourselves, how could AI improve this process of metadata capture for the submission authors? And so we've developed an AI suggester within this tool. So for this tool, the submission content is fed into an AI, like the study description. I called out study details when I first started presenting and promised that it would be important, and here's why. The AI uses this free text within your submission that describes your study, what your samples are, where they were collected, what type of data was generated, and what science questions might be of importance for your research to then generate metadata suggestions and guidance. So that example study that I was just showing resulted in an AI-driven metadata suggestion that the submission author should fill out mean annual precipitation. We really want to be transparent with submission authors about the suggestions and give control to the author for whether or not suggestions are implemented and whether or not metadata is from the AI is used. And so we are very clear about AI suggested metadata fields, and we require that the source or the AI's reasoning be provided within the suggester tool. So here the source is saying that the study description investigates chronic drought stress and reduced irrigation, making annual precipitation highly relevant. And so then it's up to the submission author to determine whether or not they should fill this out. So with that, we allow you the ability to dismiss or address these recommendations at your discretion, because we all know that it can result in some maybe incorrect recommendations. And so it's at the control of the submitter. This example that I'm showing here is using the free text in that study description that I showed. In addition to free text, this tool can also reference publication, award, and data DOIs. So it has a way to go out into the internet and look for that relevant information, pull in the details that are provided by those DOIs and by that text, and tailor your metadata suggestions so that it's more specific to the experiment and the samples that you're describing. We are building out this UI to also improve, or not improve, sorry. We are building out this UI to also provide metadata value suggestions. So what I'm showing here is explicitly just we think that the AI thinks that this metadata field is important for your study. We have the functionality also to say, based on other information, this is what you should fill out, similar to the elevation tool that I showed earlier, but using AI guided suggestions and being able to provide additional suggestions. This tool will be available in the AI soon, or this tool will be available in the UI soon. The metadata suggester tool, in summary, provides autocomplete and targeted metadata capture, lowering the burden on the submission author and allowing the individual to be able to be more targeted with the metadata fields that are important. This functionality is pluggable and can be applied to other stages within the data lifecycle. So, for example, we are applying this tool to bring in additional data from other public sources so that we have some additional information and context that we can share. And again, being very transparent about AI guided metadata versus human curated. With that, we're also developing an agentic framework to support this tool to improve the metadata suggestions and guidance. So again, continuing to just expand on and improve the experience for metadata submitters and submission authors to know what they need to capture. Now, that was all about standardized metadata and why it's important. And so I will pause here and ask if there are any initial questions for me about the submission portal or the AI tool, or we can take those at the end. Okay. I will go ahead and hand this off to Alicia to talk about data access.
Okay, hi, my name is Alicia Clum. I am the data portal product owner and the QA/QC manager for the whole project. So I'll be talking about a couple of different ways that you can access NMDC data. So I'll be talking about NMDC's data portal and then our API and Python packages for programmatic access, and then accessing data within Kbase's Lakehouse. So this is our data portal. It's a UI for accessing multi-omics data. We currently host over 70 studies, over 16,000 samples, and over 100 terabytes of processed data. I've included the QR code here, access the URL and documentation. We have monthly releases, and after that, we have a round of going through our documentation. So we make a strong effort to make sure our documentation is up to date for all of our tools and the workflows. I just wanted to run through some of the basic features that we have. So on the left panel, we have a faceted search. So you can search by things like functional terms, PI, information about samples, habitat information, where the data was processed, some of the workflow information, etc. So there's some good options here. Whatever search criteria you've applied shows up in the top left here. So this is what the active search term is. You could search by the data type. We use ORCID for login and authentication. Anyone is allowed to come to our portal and search, but if you would like to download data, you do need to be logged in with ORCID. We also have features like a map to search by geographic location, an upset plot to look for samples that have multiple data types, and you can search here by collection date, sample collection date. Our central organizing principle for the data portal is studies and samples. So you can click on these boxes and this will apply a filter for just the study. You can click on these arrows and it will take you to detailed pages about either the studies or the samples. You can click on these boxes and expand and see the workflow results and download individual files. If you're interested in bulk download, you can use these options right here. And so you can use this to download both files and metadata about samples, which we'll talk about more in a couple of slides. This is an example of what our sample pages look like. So it includes a name and description of the sample, some basic attributes like location. We describe where the sample was collected from. We link out to alternative identifiers in other systems. And something that's new is being able to download the sample metadata directly. So this would be if you just wanted to download the individual sample, you'd click here and it would download a JSON file with the sample metadata. Similarly, we have study pages. A couple of months ago, we went through a redesign, which we hope is streamlined and simplified the study pages. So we have title, description. You can click on these buttons and it will take you back to the main search page with that filter applied. You can get information on who's involved in the project, links to publications, information about funding, things like that. And then similarly, you can now export the study metadata directly here. We have a couple of new features that we wanted to walk through. So now you can download metadata as well as the data files. So you can click on the download button and you can click on the data products and select the workflow or the individual file types that you want results from. And you can click on metadata and select what types of records you would like metadata for. And just a reminder that this does require login with ORCID. Another new feature that we're pretty excited about is we've just released free text search. So in this example, I'm doing a search for persist, which is in part of the name of one of the science focus areas. And you can see here that it pulls up the science focus area, studies that are part of that science focus area, and then some of the samples. I did just want to note that we have placeholder parent studies for all of the science focus areas. So if people wanted to go and check out the science focus area that they work for and make sure that those pages look good. And then, we would be ready when you have data for individual studies to properly connect those with the science focus area. The same feature can also be used to search for an expanded set of NMDC identifiers as well as identifiers for other systems. So in this example, I am searching by an ID, a sample ID from the gold system. And you can see that it pulls up the correct single sample for that. Specifically indexes identifiers, we've done special indexing on this, so this should be pretty performant. And if you want more details about text search and what fields are supported, that's described in detail in the documentation link that I had a couple of slides ago. Next, I'll move to talking about our API and our Python package, which are available for programmatic data access. So we call our API runtime, and it has several different endpoints, including a bunch of very flexible endpoints. I've included the QR code and the URL and documentation. Some of the feedback that we got the last time that we did user research is that more examples would be great and additional documentation. And so we've worked really hard to try to address that. So this is an example of running a query against one of our most flexible endpoints. So you can use this endpoint to basically query any collection that's defined in the schema, any field that's defined in the schema. So we've added inline documentation to the endpoints. This is the Swagger UI view. We have direct links to where you could access the schema documentation if you're not sure how to query. So for this endpoint, we're doing the same thing that I did a couple slides ago, where I'm trying to search by a biosample identifier. So here I'm specifying biosample set as the target collection. And then I'm searching by the field where I expect this, and then the identifier. And then it will return a JSON in response. So you can download this from the Swagger UI, or you can, run this in a Python script. There's a bunch of different ways to access this. A lot of our endpoints don't require a login, so that's nice too. There are a couple more advanced ones where you would need to ask for special credentials, and you can also use ORCID to authenticate here. We have provided a Python package to help interact with the API for things like, automatically paging to get all the samples. And you can install this with pip. So this is publicly available. We have spent a fair amount of time working on a series of Jupyter notebooks in both R and Python, which demonstrate how to use both of our API and the utilities package. So I've included the repository. This is a great place to go if you're unfamiliar with what endpoints you should be using. So this is on top of just the basic documentation endpoint. So these are really good references if you want to, figure out how to access our data programmatically. Okay, so lastly, I'll talk about how you can access data in the new K-Base Lakehouse. So K-Base is going through a transition period right now. So they're moving from a narrative-based application to using a Lakehouse is a different kind of database. And then moving forward, they'll be using this Lakehouse and then agentic AI to help users analyze data. You may have heard about this at the BSSD PI meeting. There was a workshop. And so we've integrated with K-Base to make sure that our data can be provided to users. So this is what it looks like. I just want to note that this is not publicly available in K-Base yet. It is available for testing. So if you want access, you would email the K-Base team. If you were at the BSSD PI meeting, at that point, K-Base was pulling our data, and we've switched to a model where now we're pushing all of our data in. The technical term for like the namespace where the NMDC data is called a tenant. So you would ask for access to NMDC's tenant within the Lakehouse when you request access. So I just wanted to walk you through what some of the databases within our tenant look like. So metadata here is where all of the NMDC metadata lives within the K-Base Lakehouse. And then the results would be all of the workflow results. So there's a couple other databases, but we just wanted to orient you a little bit. So you could use this to do more advanced analysis that might not be available in our portal. For example, if you wanted to do metabolic modeling or compare your data to the rest of the data in NMDC, that this is where you could go to start doing some of that exploration. All right, we just wanted to acknowledge the great team. We have great staff from a couple of different labs, and we've talked about today metadata capture and data access, and we've included some ways where you can get involved and follow us.
So finally, we are launching a new initiative, NMDC Connect, which will be our version of standing drop-in hours. This is being done to increase accessibility and product support for both you and your research. We will be available from 10:30 to 11:30 PST every 2 weeks, starting on June 5th, and we look forward to seeing you there.